the recording on so that the students who are not there can have a look. So the course outline will talk about prokaryotic and eukaryotic genome structure and their organization. So here we are going to discuss in detail what is a genome and what are the differences in the structure of the genome of prokaryotes and eukaryotes, and this will not only include the structure but also, I mean, in terms of their organization, like how such a long genome, which is several meters in length, can be packaged in a small 2 micron bacterial cell or in a eukaryotic nucleoid. So we are going to learn this part. The second part is, and in the first part, we are also going to discuss how we can visualize the genome, whether it's visualization in a bacterial cell or a eukaryotic cell, such as mammalian cell. How can we really visualize the genome? How can we really study whether there is aneuploidy, whether there are extra chromosomes, whether there is duplication, because all of these are going to have consequences. For example, people have 6 fingers, so why they have 6 fingers? So we are going to discuss all of these in the first topic. The second topic is genome analysis. So here, when we talk about genome analysis, we are going to discuss how we can analyze the genome. So here, what I mean is that we are going to discuss the basic principles of isolation of the genome and then its analysis in terms of, for example, if you have a Saccharomyces cerevisiae, which has, for example, 16 chromosomes, right? So if how can we analyze whether it has 16 chromosomes? If you have an unknown cell, how can we analyze how many chromosomes are there? How many? What is the total genome? Then what is the ploidy, like whether you have one copy of the chromosome or you have multiple copies of the same chromosome, or they are all different. So how can we do this analysis? So this will be covered in genome analysis, and this will also include the various techniques, for example electrophoresis, which will not include simply Agarose gel, but we are going to discuss the various variants of electrophoresis for the separation of nucleic acids, whether it's a small oligonucleotide or it's a plasmid or it's a genomic DNA, right? So what kind of electrophoresis variants are available for analyzing that? So that will be the component of the second topic, which is genome analysis. The third topic is restriction enzymes. So here we are going to discuss the need of restriction enzymes, and then the various classification, the various types of restriction enzymes, and then the applications of these restriction enzymes. Then we will also discuss PCR, which is polymerase chain reaction. Again, the basis, the basic principle, and then we will discuss just 2 or 3 variants of PCR, because you will learn more variants of PCR in another course, which is recombinant DNA technology, which you will do next year. After this, we are going to discuss mutagenesis. So mutagenesis, as the term itself says, that any variation from the normal sequence is actually mutagenesis. We are going to discuss the various methods of mutagenesis, which can include chemical mutagenesis, or it can include, for example, introduced mutagenesis, which can be site-directed or random. So here we are going to discuss the various methods of mutagenesis in detail, and in this section only I'm going to cover mutations also, like point mutation, silent mutation, temperature sensitive mutation. What are the various types of mutations, like lethal mutation, conditional lethal mutation? So we are going to discuss this in this section only. So we will call it as mutation and mutagenesis. This will be followed by gene cloning. So here we are going to discuss the various methods of gene cloning. I'll be introducing you to certain other enzymes such as ligases, polymerases, etc. So we are going to discuss how we can clone a gene. This will be followed by the various methods of gene transfer in bacteria. So here I am going to talk about microbial genetics. So microbial genetics would include transformation, conjugation, and transduction, and again in this we are going to discuss in detail how these three techniques like transformation, conjugation and transduction can be used not only for introducing the plasmids per se, but how can these three be used for genome engineering in bacterial cells. How can we actually make a cell, make changes in the genome of the cell. So we are going to discuss all of these, the various methods of transduction, for example, generalized transduction, specialized transduction, which one to use when you want to introduce a gene. So those things we are going to discuss in detail, and I'll try to give certain examples for all of these. So what I try to do is that whenever we are covering a topic, I'll try to give real life examples so that you can get the concept, and also you have a feeling of the applications of these concepts. Towards the end, we are going to discuss DNA replication. So DNA replication would include not only simple chromosomal replication, but we are going to discuss plasmid replication as well. The various modes of replication like theta mode and rolling circle mode, the mechanism, and then most important is the regulation of replication. So how a bacterial cell doesn't keep indefinitely replicating. I mean, why the chromosome makes 1 to 2 copies, why it doesn't keep firing and keep making more and more copies. So that means there has to be some level of regulation, right, which is controlling that. Okay, now you have made 2 copies, now you should not initiate any further replication. So there comes the role of regulation. So we are going to discuss the various mechanisms which are developed for a regulation of replication. Then we are going to discuss transcription, which is like synthesis of RNA from DNA. So how transcription takes place, and translation is protein synthesis. So from messenger RNA, how the polypeptide chain is formed, and then how a protein is folded and uh, is used for its functionality. But again, we are going to discuss this in terms of mechanisms as well as regulation. So transcription and translation are also highly regulated. So we are going to discuss how these are regulated in the section called gene regulation, because it's the genes which are transcribed and translated. So that is why I have made a separate section for this, and we are going to discuss the various models, for example, lacoperon and tryptophan operon. So one is a catabolic operon and one is an anabolic operon, and then we are going to discuss their organization, their functionality, and the mechanism of regulation. So ideally, students, the course outline, the two colors that you are seeing is the one which is in red. I'll try to cover at least the red part before minor, and then the black part will be the syllabus the black part will be the one which will be covered after minor. So usually this is the case, but in case we are uh, able to cover more, uh, we can cover more. So it completely depends upon how you people are responding. So as I said, the class has to be interactive. You need to tell me whatever you are following, whatever is not clear, and if students are following, then we can cover all of these topics, and then we can try to cover certain switches which are called as oscillation switch, toggle switch. So these are like genetic switches which are used a lot in synthetic biology. So depending upon how much we are able to cover, we will try to include that. Otherwise, for those of you who are interested in these topics, you can do a self-study later on, or we can have a class later on. But from my side, this will be the course for sure, and depending upon how we progress, we can add genetic switches. Any suggestions, any recommendations from this part? Do you want me to add or delete any topic? Class, as I said, you need to tell me. Otherwise, I
Ma'am,
will.
I guess nothing as of yet. Or maybe once we go through the course, so maybe we'll have some topics that we need to get added.
Okay, okay. But then do you find any topics familiar or they are all new?
No, ma'am, there are quite a few topics are familiar.
Okay, okay. So then what I suggest is that maybe you people go over the topics which you think you are familiar with so that we can quickly have a kind of quick revision and then we can move further. I mean, we can discuss those in depth and then the basics can be quickly revised. Is it fine, class? Udita, Dev, Priya, Dhwani, are you people there or just?
Yes, ma'am, it's fine.
So then the suggested books are
Yes, ma'am.
okay. So the suggested books are Molecular Biology of the Gene by Watson, Gene Cloning by T.A. Brown, and then I'll be referring to selected papers from journals, and these papers I will be uploading on Moodle and then you can actually download them. So for example, I will be referring to research papers for genome structure. I will be referring to research papers for PCR and for restriction enzymes and for certain other topics because there are some topics which are not yet covered in books in that great detail. But then I will make sure that the topics are clear to you. The mode of teaching is going to be synchronous on Tuesday, Wednesday and Friday from 9 to 10. I'll be using PPT, so then you need to be there in class in the time. I will be uploading the recorded videos on Impartis, but because in Teams they are available only for a few days and after that they delete it. I would suggest that you join early. I mean, by 9 or 9:05. So I am like I try to turn the teams on at least 5 to 10 minutes before 9 because then if the entire institute will turn the teams on at 9 then I feel that there will be problems in logging in, so I usually sign in before. And my experience is from the last 1.5 years that students are almost always there in time, and I expect that your batch will also be there in time, your class. And so whenever we discuss a topic, so usually students, if they have some difficulties, you can discuss either before the class or after the class. So I'm usually there like 5 to 10 minutes before 9, so you can join in early and discuss with me, or you can discuss after the class. Or if you think that I mean there are questions which are specific to an individual, then it can be answered before or after. But then what I suggest is that let the class be interactive, and I do not want to see your hands raised. You just unmute yourself and ask me directly. Tests and evaluation, so you need to be sincere. Utmost sincerity is expected, and penalty can be imposed, so you need to be careful in that, right? uh, the evaluation, so minor one will be of 30 marks. The timings will be as per the institute calendar. I will be allotting you a term paper, and this will be for 15 marks. We can keep the tentative date after the mid-sem break, although in these online semesters you people do not get a long mid-semester break, but still I thought it will be better if you have some gap and then you can prepare well. When we talk about term paper, what is expected is that you should not be copying and pasting. So whatever you write, you need to maintain originality. We will have two viva voce examinations. So these are going to be like oral quizzes, and one I thought we can have it after minor one, and this is only for your ease of convenience because you will anyways be preparing for minor. So if we keep the viva immediately after minor one, I guess you should be well prepared, right, class?
Yes, ma'am.
Yes, ma'am.
Okay, and then for Viva 2, I thought we can keep last week before major exam, because in any case you will be preparing for major exam, so no extra effort. So the pre-major total will be of 65 marks, and then we will have a major exam of 35 marks. The timings will be again as per the institute calendar. So hope this distribution and the evaluation components, hope this part is fine with the class.
Yes, ma'am.
Everybody?
Yes, ma'am.
Yes, ma'am.
Okay, then about reminer. So if in case you are missing, I mean if you have some health issues, so then I suggest that you submit the medical certificate within 2 weeks. So you can inform me by email that yes, you will not be able to appear in the minor exam, so that we can conduct the reminer quickly. And then for any queries, any clarifications, if you think that there are too many questions and you are not able to discuss during the class or before the class or after the class, then feel free to email me, and then I'll try to answer the doubts either individually, or we can, like last year, I will tell you there were students who would email me a list of doubts, and then what I used to do was before minor and before major I used to keep a doubt class, and then every student would bring their doubts, and then we all used to discuss and interact. So that is how the course had been running so far. I will be trying to just to make yourself comfortable. I'll be showing you in between videos also wherever possible. Unfortunately, with Molbio this time the lab component is not going hand in hand, because always what I try to do is whatever is taught in theory, students used to do it in the lab, so then it goes in the long term memory. But this time the practicals will be held hopefully in the month of December. So what I will do is I will be showing you the quick videos which you can, depending upon the time, I can show you in the class time, or otherwise I will share with you the link so that you can observe the videos later. So that is all from my side for today. This is all about the course, and we will be starting with the regular class from tomorrow, starting with the genome and genome structure and organization. Um, so any doubts or any questions or anything which you want me to include or exclude, feel free to tell me. And while you people are thinking about and telling me, I will quickly have the attendance of the students who joined late. Arish Khan, Abhinav Kumar Singh, Anand Vardhan, Ankit Raj.
Present, ma'am.
Ankit Raj, okay.
Yes, ma'am.
Ashu, Abhi Agarwal, Prashant.
Yes, ma'am.
Prashant, are you there?
Yeah, yes, ma'am.
Okay. Jayesh, Chaudhary Jayesh.
Yes, ma'am, present.
Okay. Gir, Verma, Harsh, Manshu Anuragi, Pavan Nayak,
Present, ma'am.
Ashish Jain,
I'm Krishwa Kashish.
Kaushik,
Present, ma'am.
Kaushik Das or Kaushik?
Kaushik Das, ma'am.
Okay. Madhukar, Mayank Dhaka,
Present,
Yes.
ma'am.
Present,
ma'am.
ma'am.
okay. Naman Singh, Anas Chaudhary, Prakshi Gupta, Pratham Bansal,
Yes, present.
okay. Radhika,
Yes, ma'am.
Yes, ma'am.
Rajat, Ratnesh is there, I saw him. Ritika,
Yes, ma'am.
Rudransh Chaudhary.
Present, ma'am.
Okay. Shresht Saxena, are you there or no? Shresht, Shubham, Avik Singh,
Present, ma'am.
Vivek Sehra, Yatik Singh,
Yes,
Abhishek.
ma'am.
Okay, Yatik.
Yes, ma'am.
Okay. Abhishek. Okay. So I think that's all from my side today. So please tell me if you want me to add any topics, or otherwise as we go along we can discuss more and more. And so we will see.
Excuse me, ma'am.
Yes,
Ma'am,
Vishanta.
can we have a few classes on CRISPR?
We can have a few classes on CRISPR, yes, maybe towards the end. But CRISPR will be discussed actually, Vishantan, in two courses. I think RDT, I'm not too sure whether Dr. Ritu covers or not, but we have a dedicated course called Genome Engineering. So genome engineering includes all possible genome engineering techniques like Kree-logs, FLIP-FRT, CRISPR, I mean whatever you think, zinc finger nucleases, talents, everything is covered. And it also includes next generation genome engineering technologies. So CRISPR we will be discussing a bit when we will be discussing about the immunity and adaptive response of bacteria, but then I guess you are interested in how CRISPR is used for engineering the genomes, right?
Yes, ma'am.
Right, so we will see how the course progresses. So depending upon how it progresses, maybe towards the end, I will take a note of it. And so I have genetic switches and then CRISPR. So depending upon how we progress, we can include. Any other topics?
Sure, ma'am.
Yukti, Agrim Sharma. So some of the names I am able to remember.
No, ma'am.
No, ma'am, like not at the moment.
Okay, then I think.
Ma'am, I have one doubt. Like, for tomorrow class, can you just tell us which chapter will be it will be on from the book?
Okay, so that's a good question, Agrim. So that's the problem. So whether the class wants bookish knowledge or you want updated knowledge. Let me ask you this question. Whether you want just whatever is there in the book or you want like ekdam up to date.
No, ma'am. Basically, like, just go through. Like, for tomorrow class, we will just go through that topic and just attend the class. It was like that only.
No. I got your point, Agrim. So what I'm trying to tell you is that prokaryotic genome structure and organization is not there in any book. Okay, and eukaryotic genome structure and organization you will find in J.D. Watson. So J.D. Watson covers I do not remember the exact chapter number, but then you will see the chapter on genome packaging, which includes bead on a bead model, solenoid model, histones. So genome packaging in eukaryotes is covered nicely in Watson, but nobody cares about prokaryotes. They have not discussed even a bit. So that part you will not find in the textbook. So the way for tomorrow's class, we are going to discuss basically what is the genome, what are the variants of genome that the class is aware of. For example, what is chromosome, what is chromate, what is plasmid. Do you know what is chromate? This has again not reached the textbook. Agree?
No, ma'am. No idea about chromate.
Dipanshu, I will note down your attendance. Agrim, yes?
No, ma'am. I do not have idea about chromate.
Chromate, yes. So again, because it has not reached the textbook. So, but I guess the class must be aware of chromosome and plasmids, right?
Yes, ma'am.
Right, ma'am.
Right, so that is what. So what I will do is that we will start from the basics, and whatever new topics are there which are not there in textbooks, I will keep telling you in between. So tomorrow's class will be mainly on genome, genome structure, genome organization, genome shape, genome size, genome ploidy. We will discuss just this part. And
Fine, ma'am.
slowly I will take you to how they are organized, and whichever part is there in textbook I will be mentioning that.
Okay, ma'am.
So it is very easy to include, for example, whatever is there in the textbook, but then I thought then you will not have एकदम up to date knowledge. You will miss out many things.
Yes, ma'am. Up to date knowledge is better, I guess.
So there are research papers, but then they have not reached the textbook. There are some very latest textbooks, but then again they have covered some of the topics, but not everything is there. Like for example, regulation of replication, you will not find any textbook talking about that. But then this is so important that, okay, you cannot keep replicating, right? You need to have a control. So understanding the control is equally important.
Yes, ma'am.
Ma'am, I had a question.
Yes.
Ma'am, so is there any attendance policy in the course?
Attendance policy in the sense?
Like, is there a minimum, like, so for, is there a 75% attendance clause, or otherwise you get a grade down?
Grade down? No, I'm not going to do grade down for sure.
Ma'am, so that is what, because you...
No, so Dev Priya, I'm not going to end your entire class. I never do grade down in any of my courses, whether it's a 700 level course or a 200 level course. But then I do take attendance, because then I would like to know who is participating in the class, just for myself.
Okay,
like
that is what I wanted to ask.
This is just for knowing you people, that's what, because in this kind of online semester I'm not able to see you, so at least I would know your names, recognize your voices. That's one way of doing that. That's it, nothing more than that.
Okay. All
But
right.
then in the long term you will realize that the students who attend the classes, they always understand the concepts better, for the simple reason that when I'm explaining you the concept, you can ask me questions then and there. If you are listening a recorded video, then it's completely different, right? It's like reading.
Yes.
When you are sitting in your own office, in your own room, isolated. So many students like that, but then I guess my experience is that my students are always, the students always used to attend the classes and used to interact with me, and I really like that. Let's see how your batch goes.
Yes, ma'am.
Ma'am, one more thing. Ma'am, the course you mentioned, genome engineering, I don't think it has been floated in the past few semesters.
Genome engineering?
Yes, ma'am.
Yes, so genome engineering, Vishantan, has a lab component, and since the online semester, we haven't floated for the past 2 years, for the simple reason that I do not want this course to be running without lab.
Right, ma'am.
This course is something which is a lab heavy course. It's a 202 course, so lectures are only 2 per week, but then it requires lab component, practical, hands-on.
Ma'am, so is it possible, like, if at all the next semester is offline, so would it be possible to get this course
Yes, it will
floated?
be possible, right? I have received enough requests. We will float it.
Okay, thanks a lot.
The reason, Vishantan, initially I wasn't floating was that the combinatorial biotechnology, so I offer this course called combinatorial biotechnology in the semester 2. So this course was also, like, many students used to tell me, ma'am, you offer combinatorial. So that's why genome engineering was not floated, but then another reason was the lab component. But let's see how the situation is next semester.
Okay,
I have
thank you.
floated genome engineering together with mol bio. a few times because then both have labs and but then let's see.
Okay, ma'am. Thank you.
What about others? Any comments, any suggestions, anything that you want to say? Anything about evaluation policy? Ratnesh? Rudransh? Everyone is quiet. Ritika? I have left already, I guess, right?
No,
Oh, no, ma'am.
Yes.
If Priya, you were saying something.
No, I just said that no, we are here.
Okay. So then should I close the meeting? We will start the regular class from tomorrow?
Yeah, sure.
Sure, ma'am. Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.